Okay, we'll get started. A warm welcome to everyone. Welcome to today's webinar. Our webinar today will be on the pathways and opportunities for a career in vaccine research and development. This webinar is the first in a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. Let me just have my camera on for introductions. And I hope everyone can hear me. Can hear me and see me. My name is Joy Kiptim from the KEMRI-Wellcome Trust Research Programme, and together with my colleague Shadrach Baraka will be the co-facilitators for this session. Shadrach, you can go ahead and introduce yourself.
So as you've heard, my name is Shadrach Baraka. I'm an Assistant Research Officer in the Schools Engagement Program. My role is community engagement, working with young people to promote interest in science and science-related careers. Thank you.
Thank you, Shadrach. Shadrach will be back later on to take us through the question and answer. But before we start this session, a few housekeeping matters. The first is this session is being recorded. Your personal data will not be publicly available, and you'll only use this to analyze the impact of this webinar and see ways of improving this webinar. For those who are not conversant with Microsoft Teams, kindly take note of the chat box option. If you're using a laptop, this should be at the top of your screen, but if you're using a phone, it will be at the bottom. The icon of the chat box platform is a word bubble. Kindly feel free to use this option to share your questions during the presentation, as well as any other thing you'd like addressed. We'll have a few slots open for people to directly ask questions to the presenter. If your internet is stable, you can raise your hand using the available icon, which is written raise. It's a palm. For quality control, we have disabled your microphones and camera, and we'll only be able to enable some of your microphones during the Q&A session. Now that we know where the chat box option is, we would like to hear from you. I've posted a link on the chat section. It's a pre-survey. Kindly take a few seconds to fill this out. It takes an average of 25 to 30 seconds to fill this out so we can be able to know your expectations and where you heard about this webinar. So I'll take a short break for you to be able to access this. Okay, I see some of you are already responding. If you're having a struggle accessing the link, you could also let me know. And if you're done, you can give me a thumbs up using the React so I get a cue to proceed. Okay, I see a few responses coming back. In the interest of time, I will go ahead and just give a brief background of the KEMRI-Wellcome Trust Research Programme. Feel free to fill in the survey even as I do this. So the KEMRI-Wellcome Trust Research Programme is a world-renowned health research unit of excellence based in Kilifi and Nairobi, Kenya, as well as Mbale in Uganda. And increasingly, we're involved in regional collaboration to support research in the neighboring countries. We have over the years excelled in the use of novel ideas, working with local communities to achieve better health for Africa while also developing African scientific leaders. Our vision is to undertake cutting-edge and novel research relevant to national, regional, and global needs. Within the program, we have the Schools Engagement Program that works within the school system in Kenya. We have programs engaging with students within primary, secondary, as well as universities. The University Engagement Platform provides a safe space for young people to engage with researchers. The goal of this engagement is to enhance understanding of research, empower the next generation of African research leaders, and provide a platform for dialogue. Over the years in Kenya, we have engaged with young people from different institutions on a wide array of health and health-related dialogues. Today, this webinar is part of our university engagement initiatives. For our webinar today, we are glad to have Professor George Warimwe from the KEMRI-Wellcome Trust Research Programme and Professor Teresa Lambe from the University of Oxford. In this session, we'll have 2 presentations and a question and answer session. We hope you'll find this engagement informative and engaging, and you'll be able to learn something from it. With that said, I would like to now welcome Professor George Warimwe to give us a brief background on the Vaccinology Webinar Series and then go ahead with his presentation. And even as he comes, for those who are joining us, please ensure you give us feedback so that we can get to understand what is the expectation and see how we can improve this engagement. Over to you, George. I see your presentation has loaded. You can go ahead. Karibu.
Thanks a lot, Joy. Can I just confirm that you can hear me?
Yes, I can hear you well.
Great. So thank you all for joining this. So as Joy mentioned, I'm George and currently Deputy Director of the KEMRI-Wellcome Trust Research Programme, which is based in Kilifi. I'm also a Professor of Vaccinology at the University of Oxford in my other hat. So this program that we're going to describe is around basically tapping into African talent for the purpose of providing solutions to global health. We've got lots of challenges in the world today, things like climate change and antimicrobial resistance. Everywhere you look, there's lots of things that need solutions at a global level. And my view is that until the talent in Africa is actually engaged and involved, that we are really not going to be able to address these global problems sufficiently or in a way that is sustainable. And so from this angle, I've been thinking more around how this talent in Africa, of which there's a lot of it everywhere you look, can be harnessed for the purpose of vaccine development from a very early stage, so that we're actually providing these solutions to the world in a way that is different from what happened during COVID. So I'll jump straight into it. I'm sure you've heard about vaccines. The last few years, because of COVID, there's been quite a lot of talk in the media and everywhere about the value of vaccines. One of the things that's really troubling for us as a continent is that we produce less than, I think it's actually less than 1% of the vaccines we use here. So, children get immunized during the first years of life to protect them against some of the most common illnesses. And these vaccines have had a massive impact in the protection against these illnesses. For example, measles, yellow fever in some countries, and so forth. And this impact of vaccines, also extends to animals. We've had 2 diseases eradicated in the world. The first was smallpox, which some of you may have heard about. but the other is rinderpest, which was a disease that was had major economic impacts to farmers in Africa. And the value of vaccines in doing those, it has been immense in terms of eradicating those two diseases. And there's hopes now for having a significant dent on the burden of other diseases through vaccination. Malaria is a good example, and we've heard about new malaria vaccines that have been approved in Ghana, and there was a previous one also from the GSK. So vaccines are becoming, they've always been important, but there is realization of their importance in a country's and a continent's security, public health security. And so the African Union has then therefore set this goal to increase capacity for vaccine production, research, and development on the continent, from this situation where less than 1% is produced on the continent in 2021 to a situation where at least 60% of the vaccines are produced on the continent. This is for our own needs, but also for the needs, we can sort of export these vaccines to other countries. And that's what this slide is all about, that vision. Now, to support this, there's been lots of investments coming through. And again, you may have heard it from different media, and this is just a slide from one of the white papers that have been published, talking about 30 African vaccine manufacturing projects announced across 14 countries in Africa. And this immediately creates opportunities, but also highlights the gaps that are there. And one of the gaps that have been highlighted is the need for a workforce that is actually knowledgeable about vaccines, motivated to work in vaccines, and to work within an ecosystem and build that sort of ecosystem that would support a thriving vaccine R&D sector on the continent towards those ambitions that I've highlighted. But it becomes immediately apparent that there are not very many opportunities to learn about vaccines on the continent, at least. And the programs that are there are either not sufficiently detailed or relevant for the different facets that are required for a thriving vaccine R&D sector. And the other element is that the programs that maybe there are not really supporting, preparing the candidates that go through that for the job market. So, for example, the expectations of industry in a sort of personnel working in that manufacturing company, so that's what I mean by industry, sometimes do not align with academic expectations of what vaccine R&D means. And so to align those sort of expectations, there's a need to design programs that are actually fit for purpose. Now, one of the options is, we have proposed 3 strategies that we'll be taking forward over the next 3 or so years. The first one are these apprenticeships, which is basically African scientists are placed within global vaccine manufacturing companies, and they spend time there for anywhere between 3 months to 12 months, pick up skills in they're actually just being in an environment, and sometimes just being in an environment is sufficient to inspire you to do something different and also just to contribute. So this is not just about a one-way learning, but it's also contributing the knowledge we have, the contextual knowledge of being in Africa, operating in Africa, and adding value to that process so that, again, we achieve that ambition of Africa being a key player in this sector. So this will be adverts that we'll be putting out in the coming few weeks to months to identify, scientists working in Africa, students and postgraduate students, etc., and pairing them with vaccine manufacturers where they have this sort of exposure. And the vaccine manufacturers we're working with are diverse, including companies in Africa and also globally. A second work stream that we are focusing on is this vaccinology courses. So this is just an information giving exercise, what we are doing today. And but vaccinology courses will be about a 2-week session with a faculty of scientists, vaccine experts, and describing the basic concepts of vaccine R&D from what we call target product profiling, working out what are the characteristics you need in a vaccine, to the process of what it entails to develop a vaccine, to all through to deployment of vaccines in populations and assessing their impact. So this again will be happening once a year, and there are many other courses on the continent that one could target. And then finally, this is all Vaccinology Career Fairs, which is what we are. This is the first of these fairs. It's all information giving and signposting yourselves and other students, university students across Africa, of places where they can find further information about going into vaccine R&D. Sometimes any new opportunities that are arising, jobs, PhD studentships, master studentships, etc. So that, again, feeding into that ambition of Africa being a key player in this setting. So so that's all about the actual program. And we have multiple funding. We have funding from the Bill and Melinda Gates Foundation. We receive support as KEMRI-Wellcome Trust Research Programme. We receive support from the Wellcome Trust as a co-funder, and there's been in-kind support by the vaccine manufacturers. So there are many people who would really want to see this happen on the continent, and I thank you for your attendance today, because it signals an interest in this area. Now, I'll talk a bit more about myself and how I got into vaccinology. There may be some of you in the room or in the virtual room who grew up with animals, and this is just a photo illustrating what, in my view, happens in many places in Africa, and maybe let me say Kenya, which is where I am. We live very closely with our animals. And you might take a lot from this image, but for me, this boy is sharing sugarcane with a cow. If you know how, that's a precious commodity as a boy, sugarcane, sharing it with your cow. To me, that signifies quite a lot of interaction and love for these animals. But animals are important for us because they provide not just food security, there's also financial security. I went to school, to university, because my grandparents sold a cow for me to go to university. So, and there are many other stories like those. So the value of animals is huge, and this is then got me into thinking about how can I leverage this information, this linkage we have with nature through animals to provide solutions for some of the big infectious disease problems we have. And that's not just coming out of sort of thin air. This pie chart here represents the sources of emerging infectious diseases. And what I mean here is, you've all heard of things like Ebola, Marburg, all these outbreak diseases that keep popping up every so often. Well, it turns out that if you analyze all the emerging infectious diseases that have ever been reported globally and continue to be reported, a big chunk of them actually involve animals. They are acquired from animals, or animals are involved in the pathway of spread from human to human, or vice versa. And what this signaled to me was that if we are going to find a solution for these diseases, which we call zoonotic diseases because they are transmitted from animals to humans, then we need to account for the animal component of this. And my focus coming from Kenya was on Rift Valley fever. Now, if the name already betrays where I'm going with this. Rift Valley fever was a disease that was first identified in Kenya, in the Kenyan Rift Valley in the 1930s, and has gone on to be a really major problem and a priority for many organizations globally, not least, even the African Union. So a little bit about the disease, and this would generally be the first place you start when you're thinking about vaccines, but actually many other interventions. Is like, what's the problem? What's the problem you're trying to solve? What is known about it? What's the impact? And if we look at Rift Valley fever, it's a big problem in animals. It causes very high mortality. That's very high number of deaths in sheep, goats, and cattle. So all these major farm animals, which of course are associated with very high economic losses. But because of the linkage with humans, animals then become a major source of virus to humans. And this image here represents an individual who had Rift Valley fever. This is very recently in Kenya, I think in the last 2 years or so. And most cases are mild in that they are self-limiting. You might feel a bit unwell and then you recover. But some individuals actually develop severe disease, and this is very associated with very high rates of death. And you might get some complications such as blindness due to the infection. Now, the WHO and other organizations, as I have mentioned, have prioritized this disease because of its potential to cause emergencies that emerge that result from outbreaks, epidemics, just new very high numbers of new cases of RVF, and we've seen how this can be disruptive, from COVID. So at least that doesn't need to be introduced further. But one of the things that was really fundamental in thinking about a vaccine for Rift Valley fever was that if you studied individuals who had recovered from RVF infection, which is what I'll call it, these individuals tended to have very long-lived immunity, meaning that they couldn't then succumb to infection in future or disease in future from RVF. And the observation that was made there was that the people who were involved in the initial experiments of this with this virus, some were exposed and then they returned to their countries in the United Kingdom, for example, and in a period of 25 years when they were not in contact with Rift Valley fever, they still had immunity to this virus. And the key component that was involved in the protection was this molecules we call antibodies that target the virus surface, as illustrated by the green. The green are the sort of is illustrating the antibody, and the circle is the virus, and the things that are popping out are what the antibodies target. Now, this knowledge is really important because these antibodies provide protection and what they target. So with that information, you can already start thinking, okay, I need a vaccine that generates this type of immune response. And so in that vaccine, I need to have this protein component that's on the surface of the virus to stimulate an immune response in humans. And that's generally what, the process of designing a vaccine, that's what it involves. The other element for Rift Valley fever is that these observations on lifelong immunity were not just observed in humans. They were observed in numerous occasions in these sheep, goats, and cattle. I remember I said these animals are the main types of animals that suffer from RVF. And so we could corroborate these findings from these animal species. And therein lies an opportunity to actually Now that you've designed your vaccine using these proteins and you know which immune response you want to target, you can already now start thinking, okay, so then we can immunize animals and we can immunize humans and see whether we can replicate what we see in the immune response following infection. And generally, that's all I'm trying to illustrate on this figure. The figure is a bit busy, but I'll go through it as clearly as I can. So starting from the left is what you call a target product profile. And this is what you would call a human or veterinary vaccine pipeline. They are very similar, but there are some important differences. So the target product profiling, this is where using the case of RVF, you found out what the burden of disease is, as in how much disease is there in the population, using historical studies or a prospective approach in studying that during an epidemic and so forth, looking at the impact of the disease, the types of people that are affected in a population. Is it children? Is it adults? Is it pregnant women? All that. So once you have that information, you then decide, okay, I need to target a particular immune response. In this case, again, we knew the surface of the virus has these proteins that generate these antibodies, and that's what you call the immunogen. So you put that in a delivery vehicle. And we've all heard about mRNA, ChAdOx, and many others. So those are delivery vehicles, to bring this immunogens into the body, whether it's a human or an animal. And so then from that point, when you've designed your vaccine and you've done some, there are laboratory experiments, you can confirm that the immunogen is being produced in the right way, as you wanted it to. You can then proceed to this proof of concept studies in laboratory animals. So these generally tend to be small animals such as mice, rabbits, sometimes non-human primates. And this is the first step to look at whether this vaccine construct that you made in the lab can already start to elicit the immune response that you are looking for. And if we then just stick with the human phase, the human pipeline at the top, following a period of getting good proof of concept that your construct is working, you would then go into this phase 1 trial. So there are several phases in a clinical trial. You have the phase 1, which usually is the very early, and you evaluate this in few numbers, primarily looking for safety. And then as you progress, you go into phase 2, which might have a couple of 100 people, and then phase 3, which is much larger, thousands in many cases. And afterwards, you then need to evaluate after the vaccine has been licensed. You can then do phase 4 studies to look at, okay, so now when you deploy this in a big population, in a whole country or a whole region, a whole continent, the whole world, you can then start to look at additional safety, like the rare side effects that may be associated with this that you can only see in a very large population. So that would mark the human vaccine pipeline, and you can see historically it has taken anywhere between 10 to 15 years with and that's the cost over $1 billion to get it from up through that scale. Obviously with COVID, this has been so accelerated with the efficiencies in throughout the whole process, but I mean, I think the timelines will keep dropping, at least for the human vaccine pipeline, following the learnings from COVID. And now if you then look at the bottom, from a veterinary perspective, you start with the immunogen design, and very quickly you can already progress into the target animals, so the animals for which the vaccine is intended. And you can see that you've sort of jumped this preclinical study phase because of the possibility of doing this early study in these animals, and that obviously has an impact on the timelines. It has an impact on the cost which is a fraction compared to the human vaccine pipeline. But the other processes, the process that you need to go through to manufacture your vaccine at very large scale are similar between the humans and the veterinary. At least the principles are. The principles of monitoring safety at an individual level or at a population level, again, are similar. And so too are the regulatory, the ethical issues that or animal welfare issues that you need to go through, and regulatory affairs requirements. So there's lots of synergies between the two, and these are the sort of things I had in my mind when I was working on this Rift Valley fever vaccine program or taking it forward. And the long and short of it is that I developed this vaccine. It's called ChAdOx1 RVFs. You may have heard of it previously. It utilizes the platform that has been used by the Oxford AstraZeneca vaccine, and it's highly safe. We showed that it is very safe in livestock, and the studies were actually done here in Kenya. It's highly immunogenic, means that it generates a very high level of those antibodies, that immune response that we were really interested in generating, and it provides 100% efficacy. Now, what this means is that if you expose these animals that have been vaccinated to actual live Rift Valley fever virus, they are completely protected. And this was really exciting. And so this product is now going into further development for commercialization. Because as I said, it's the same virus that affects humans and animals. The concept of a construct, a ChAdOx1 RVF construct, is one that can be applied for both livestock and humans. And so this is exactly what we did. And actually, we've recently just published some data on the evaluation of this vaccine, of course, manufactured in a different way from the veterinary, although the concept is similar, and shown that the vaccine is safe in humans. This is a small phase 1 study, as I mentioned, the size, the aim is to look at safety. But even in these small safety studies, we could already see that we're achieving very high levels of immunity following vaccination. And these levels of immunity, how quickly we are able to generate this, meets the WHO, the World Health Organization criteria for products that are intended for responding to outbreaks. So you want something that you can manufacture very quickly. And then once you immunize people, you want the vaccine to actually generate an immune response very quickly. And that's what we've shown here. There are other benefits, such as the fact that it doesn't need very cold storage. It can be kept in a fridge. And so there are many things that the vaccine is ticking. And so at the end of it, I think, of course, with further clinical trials development or evaluation, we will end up in a situation where we've provided a solution, and I feel very proud being a Kenyan and providing a solution to the world for Rift Valley fever epidemics that with a concept that can actually provide protection, not just for animals but also for humans. And of course, the impact of this goes beyond just the clinical protection in these two species. It's more also the impact on livelihoods, the impact on education, the impact on food security, etc. And of course, there's a lot of joy that animals bring us, such as is evident from this photo of this Borana girl. So that's my take on vaccines. That's my take on my sort of abbreviated career on how I got to be making vaccines. What I did not mention is that I qualified as a vet in University of Nairobi, and I've been able to do all through this, having had that background training within Nairobi University, also spent time within Oxford University and other interactions. And it's more about knowing that we as African scientists are able to have an impact in this area that is actually very, very complex, but we are able to have an impact in this area, and there are others who are having this sort of impact in vaccine R&D and manufacture. But there's room for so many more individuals to be contributing to this, and I see this as an area where there's still untapped resource from the continent, and I would really encourage you all to consider a career in vaccinology. Thank you. That's the end of my talk. Over to you, Joy.
Thank you, George. I think we can just take a few minutes to answer some questions. Shadrach, I don't know if you're ready with 2 questions before we go into the next presenter.
Yes, Joy. Thank you, George, for such an interesting presentation. The first question is from Evans, who would like to know whether there are any specific projects around vaccine research and development as part of a master's course.
Yeah, I mean, I think there'll be many. I think there are lots of adverts in different places. So universities, I mean, if you Google, you'll get different kinds of offerings around vaccine R&D. I think, yeah. So that's a straight answer to that. I think what I would like to encourage us is for us to take a perspective of thinking, okay, so there is a cost there, or there is a job there being advertised around vaccine R&D. But if we step back and think, okay, what's the solution I want to provide to my local environment, to my region, to my country? Is it something for me? It was very personal going into zoonotic diseases, because these animals have had an impact for me. You just need to identify what it is about your own sort of career. What impact do you want to have, and thinking of it through solutions. You identify the right master's program or PhD program or postdoc program for that matter. And then the value that you bring to that program is going to be a lot more. So that's something I can really encourage the audience to consider. It certainly worked very well for me, and others that I know of. So there's just some 2 cents worth of advice.
Thank you, Prof. Then we have Joel, who has a background in malaria immunology, and he'd like to know what criteria we use to reach out to grad students, and how can we support students to get training and fellowships in vaccine development so that we reach the 2040 goal. Thank you.
Okay. I think so. What I've described in our program, we will obviously be advertising some of these positions, especially the apprenticeships. And we are not the only program within Africa offering these kinds of opportunities. There are the organizations that have been funded to provide scholarships in certain disciplines. Some are very much lab focused. Others are, purely vaccine manufacturing, like our program. So I think we just need to look out. Like, you just have to follow all the right organizations. Look at which are the organizations that work on vaccines in Africa. This information generally tends to be very readily available just by Googling or whatever platform you use to look for information. And then really engaged with these organizations where you have an advert for, like I know the Africa CDC have previously announced something on training in Texas and A&M. The International Vaccine Institute also has had courses previously. So it's engaging with these individuals and really being proactive in asking, how can I join this program, or how can I improve my sort of CV to be competitive for this particular program? And most of the time you'll get feedback from those individuals because it's in their interest to sort of enhance capacity development, which is why they've gone into that field. And of course, there's more general training around research. We have a program at the KEMRI-Wellcome Trust Programme, which is focusing on just enhancing research capacity in Africa, just more generally. You may have heard of it, called IDEAL. And so there are many resources out there, and we will always be ready to assist where possible. So please do reach out.
Thank you, Prof. So we'll take one more question, then hand back to George. So we have a question from Leola from Nigeria, who would like to know what steps the African Union is taking to recruit foreign trained African vaccinologists and scientists who are interested in vaccine manufacturing. Okay.
I can't speak for the African Union, but I'm sure that they will be thinking about tapping into the diaspora. So, and I believe you mean African scientists who are based in, outside the continent. But for our strategy, would like to actually encourage, that's the whole aim of this, would like to encourage all the talent that's sort of from within Africa to basically contribute, either by being here within the continent and participating in vaccine manufacturing institutions that are coming up and developing new areas of research around vaccines that against diseases that are neglected and common in Africa. But also, if these individuals are based, say, in Italy or Germany or whatever, and they are working on a problem that's really affecting the continent, that's a priority for the continent. We don't think of that as brain drain per se, because they are contributing to a solution for the continent. Remember, it's all about thinking about the solutions to the public health, animal health, global health, let's call it that, and what you're doing to sort of have an impact in that area. And what I've been saying is that we as African scientists really need to stand up, because there is a lot we can offer in that space. And my interest is really to push this through the vaccine R&D and development, and there'll be other areas. But I think this is one area where there is potential for massive impact, and it's really untapped.
Okay, thank you, George. Thank you, Shadrach. I think I'm hoping that Professor Teresa Lambe is ready. Yes, she is. A warm welcome. You can take us through your presentation, and then we'll be back to the question and answer. Over to you.
Great, thank you. I hope you can both see and hear me well.
Yes, we can.
Okay, so I want to thank the organisers, in particular Joy and George, for inviting me here today to talk about my career and how I ended up developing vaccines against emerging and outbreak pathogens. I'm going to be a little bit selfish though. Because I have a captive audience, I'm going to try and convince everybody in the audience how important vaccines are. I'm hoping most of you are already convinced, but I like to take 2 or 3 minutes of your time and try and convince you that vaccines are one of the most important medicinal interventions ever. So this is a very old, very simple infographic from Public Health England, demonstrating the number of cases of a vaccine-preventable disease before a vaccine was introduced and then after a vaccine was introduced. But what are the types of characteristics that myself and George, and hopefully many of the people in the audience, need to think about when we're making vaccines? Well, first and foremost, we need our vaccines to be tolerable. So there's no point in designing a vaccine that induces side effects that are worse than the disease you're trying to prevent. Ideally, you want that vaccine to induce an immune response, an effective immune response that will protect against severe disease. Ideally, that immune response would be long-lived or sustained, and that immune response is typically composed of both antibodies and T cells. And one way to think about antibodies is that they are great at stopping the pathogen of getting into your body, these neutralizing antibodies, and stopping the infection before it even takes hold. T cells are particularly good at recognizing the cells in your body that a virus might have got into and destroying them. And really, we want our vaccines to induce both antibodies and T cells to protect us against overt disease. The types of practical considerations that I probably didn't consider enough when I was young and naive, but now that I'm grey and old, these are the things that I consider first and foremost. There's absolutely no point in me designing a vaccine against a disease of impact in low and middle income countries if the cost point is such that it will prohibit that vaccine getting into the arms of people who need it. So the first thing I think about now is how much will this vaccine cost before I start to design it. And some of the things that feed into the cost of a vaccine are around stability and delivery. So how stable is your vaccine? Will your vaccine have a really long shelf life because that pushes the price of your vaccine down? Does it require extensive cold chain storage? Does it need to be stored at -80 or will a fridge do? Again, a fridge will push the cost point down. And how are you going to deliver the vaccine? Are you going to give it up the nose, under the skin, or in the arm? Because again, that impacts the cost. So these are the types of things that myself and George think about routinely, and hopefully many of you will start to think about when you start your vaccine journey. The other thing that I wanted to touch on this slide is a cartoon from Edward Jenner's time. Edward Jenner is frequently referred to as the grandfather of vaccination. It's slightly misleading because both vaccination and virilation were around before his time. Regardless, Edward Jenner noticed that milkmaids who had been exposed to cowpox did not go on to develop smallpox. And smallpox, as George has already mentioned, is only one of 2 diseases that have been successfully eradicated through vaccination. It was a devastating disease. It's thought to have claimed some 300 million lives or more. And Edward Jenner conducted some very crude, rudimentary clinical trials in the late 1700s, early 1800s, and demonstrated that cowpox-derived material could protect people against smallpox. And this is a cartoon of that time, suggesting that if you were to get Edward Jenner's vaccine, that you would go on to develop bovine-like characteristics. To my knowledge, nobody has ever turned into a cow because of vaccination, but it does lend itself to a conversation we need to tackle, and probably those in the audience need to tackle it more than George and I, because we're a little long in the tooth, around vaccine hesitancy, vaccine reluctance, and the anti-vaccine movement. And I don't know that we have the right tools right now to tackle misinformation, but hopefully some of you in the audience will be able to provide insights and new innovations on how to tackle the mass information that is out there that is misleading around vaccines. As you will all know, there was an Ebola outbreak in Western Africa that started in and around 2013. It was thought to have claimed some 11,000 lives. That's a number that is thought to be an underestimate. But after the Ebola outbreak, the WHO, government bodies, and a number of NGOs came together and made a list of outbreak pathogens that they were concerned about, which might cause the next pandemic or outbreak. Many of these pathogens you will have heard of. You'll certainly have heard of Ebola. George has given a very good overview of Rift Valley fever, and some you may not have heard of, such as Crimean Congo hemorrhagic fever, although it is a disease of impact in many places of the world. And there really was a concerted effort to develop vaccines and therapeutics against these pathogens of concern. But how do we go about that? How do we make a vaccine? So for me, the first thing that you need to do is identify the pathogen of interest, the pathogen that is causing the outbreak. And when you've identified the pathogen of interest, you need to identify parts of that pathogen that you think, if you have a very strong immune response against that part, you will prevent infection or prevent disease. Routinely, the part of the pathogen that you will target is the cell entry factor. If we take SARS-CoV-2 as the example, the spike protein was how this virus got into our cells, into our body, and caused disease. And if you can stop that entry through the use of neutralizing antibodies, you can prevent infection and prevent overt disease. So what we typically do is we take the coding sequence, or the recipe, if you will, for the cell entry factor, and we stick it into our platform technologies. And two of the platform technologies that were used a lot during COVID were viral vectors and RNA technologies. So we stick the coding sequence, the genetic sequence, into these platform technologies, and then once you're vaccinated with these vaccines, your body makes large amounts of spike protein, a small part of SARS-CoV-2. And your immune system doesn't recognize the spike protein as part of your body. It recognizes it as something foreign. And in doing so, it mounts a really strong immune response against the spike protein, inducing both neutralizing antibodies, binding antibodies, and T cells. And hopefully it induces it to a level that your immunity is such that you will be protected against overt disease if and when you come into contact with SARS-CoV-2. While viral vectors and RNA platform technology were seen as the darlings of SARS-CoV-2 or the COVID-19 pandemic, there are other platform technologies that are really impactful in preventing disease both in the UK, in Africa, and elsewhere. And these are whole inactivated vaccines and protein subunit vaccines. They're used routinely for vaccination of both children and adults against really impactful diseases. And it's important not to forget about these platform technologies, nor to de-invest in these technologies, because we don't know what disease is coming next, what might cause the next pandemic. And having more vaccine platforms in your arsenal to generate vaccines against disease X is better. It may be for the next disease that a protein or subunit or whole virus platform might lend itself to inducing better immune responses. We routinely get asked, how did we manage to develop our vaccine in less than a year when it typically takes 5 to 10 years? And George has touched on this quite well, so I'll speed through this slide somewhat. So normally vaccine design, once you've identified your outbreak pathogen or the pathogen you want to protect against, can take weeks to months. It can be an iterative process. But once you've generated your prototype vaccine, you start with your preclinical studies. You start with small animal studies, and you may go on to monkey studies if there's a tenable animal challenge study that is available for you to test in. Once you've got positive readouts from your preclinical studies, it's only at that stage that you'll consider doing GMP manufacture. And that's because as you progress along this timeline, each of these steps increases astronomically the cost of the project. And really you want to make sure that the vaccine you are developing before you progress along is the best in class. So generally it's an iterative process where you go back to the start if your vaccine is not giving those types of immune responses that you want. You can do it more quickly. So the vaccine design for the Oxford AstraZeneca vaccine, I co-designed it over a weekend, and having preclinical studies being run in parallel can also speed this up. And in that way, you can help shorten the timelines from 5 to 10 years down. But this was certainly the timeline that we were looking at when we started to make a vaccine against another coronavirus, Middle Eastern Respiratory Syndrome, which is caused by the MERS coronavirus. It has a really high case fatality rate. It was discovered in 2012, and I think there's been between 800 and 1,000 deaths up to 2020. It's generally individuals that have comorbidities and are in close contact with the zoonotic reservoir. So as George was mentioning, One Health is hugely important to me and to the team here in Oxford. So we did what we do. We looked at the MERS coronavirus. We tried to identify parts of the virus that if we had a strong immune response against, hopefully would be protected against overt disease. And that was the spike protein on the outside of the virus that mediates cell entry. We did some small animal studies and some larger animal studies, and were able to demonstrate that the vaccine induced the right type of immune response, and indeed, these animals were protected against overt disease when exposed to MERS coronavirus. After these promising results that did take an amount of time to accumulate, we started a GMP manufacture with our very small on-site facility, and we started a clinical trial in March 2018. Typically, you start your clinical trial with a small number of participants, and you are concerned with safety. Safety is paramount in your mind for all phases of a clinical trial, but your phase 1 is particularly interested in looking at safety. So you typically will vaccinate one individual, wait for a number of days. If there are not overt SAEs or adverse events, you will then vaccinate 2 more individuals, again undergo a safety review, and if favourable, progress up to increase the dose of the vaccine. So we vaccinated individuals with a low, a medium, and a high dose. The safety was as expected. You generally get a sore arm and flu-like symptoms with viral vectors. We saw a strong antibody response, and we saw a strong T-cell response. And this was the trial that we had initiated before SARS-CoV-2 appeared. And when SARS-CoV-2 appeared, we really had to rethink the development process and timeline. There was no way we could take 5 to 10 years to develop this vaccine. So what we did was take a financial risk, and that is the only risk we took with this trial. In fact, this vaccine and many other SARS-CoV-2 vaccines have undergone more rigorous scrutiny across the trials than other vaccines in their development pipelines. So the risk that we took was we ran our phase 3 GMP manufacture at the same time as running our phase 1, phase 2, and phase 3. So before we knew that the vaccine was effective at protecting individuals, we were already working on upscaling the manufacture. And if the vaccine had not been protective, then this investment, this financial investment by Serum Institute, the UK government, and AstraZeneca would have been wasted. So what are the lessons that I've learned about how fast we can go? Well, first and foremost, you need to plan for the future and plan many steps ahead. Plan A is unlikely to work. Plan B, C, and D, maybe by the time you get to F, something might go smoothly. You absolutely, in my opinion, need to partner with pharma early. Academics are great at blue skies thinking and pivoting fast, but we do not have the skill sets for licensure. We need to work at risk, again, financial risk. We always prioritize the safety of our volunteers, and collaboration is key. Making sure there is open dialogue between government, regs, international bodies, charities, WHO, academics, and industry enable these vaccines to get licensure in less than a year. So a little bit about that journey across 2020. So as I've mentioned, the vaccine was designed over a weekend, and the sequence was released on the 12th of January. By the 17th of February, we had a prototype vaccine that was suitable for small animal studies. To say the team was small at that stage is slightly misleading. This is a picture of me making up the vaccine in a hut to go into these small animal studies. It was one of the few times I was allowed back into the lab because apparently I'm too messy and my writing is illegible. Regardless, we were able to demonstrate 9 days later that the vaccine did induce an immune response in small animals. And then we picked up our phone to our collaborators that could do these bespoke challenge studies. And it really was studies from the Rocky Mountain labs in non-human primates that gave us the confidence to move on with our clinical trial. So as I've mentioned, the GMP manufacture was ongoing while we were doing these animal studies, and it was released to trial on the 22nd of April, and by the 30th of April, we had partnered with AstraZeneca. And we had started vaccinating. And this is a photo of one of our 2 volunteers who came in to be vaccinated with our first doses of the Oxford AstraZeneca vaccine. And we were very grateful she came in and didn't even have to roll her sleeve up. But what wasn't so helpful or pleasant was she woke up to news stories 3 or 4 days later that she had passed away due to vaccination, which was obviously a complete fabrication. And we and many others tried to disarm this misinformation, but it's really difficult trying to get that genie of misinformation back into the bottle after it's escaped. So a little bit more about the phase 1 trial. This is not a typical phase 1 trial. The trial that I described earlier, looking at the vaccine responses in 24 people, is a typical phase 1 trial that we would run. Our phase 1 trial for the Oxford AstraZeneca vaccine had over 1,000 participants, and they were randomly allocated into 2 groups. Those individuals that would get the investigational product, the ChAdOx1 nCoV-19 vaccine, and those individuals that would get the placebo. So really what you want to do is you want to count the number of cases that occur in each of these groups. And while you never want anyone to get COVID, you're hoping to see that there are fewer cases in those individuals that have been vaccinated with your test vaccine. And there's absolutely a huge team is needed to deliver these clinical trials and the manufacturer. Sorry, my slides seem to have a mind of their own and want to keep skipping on, so apologies, I keep skipping back. The team that is needed to deliver manufacture and clinical trials is absolutely massive. And even if you don't like science, there is a place for you. If you're curious and if you're determined, there is a place for you in helping deliver key medicinal interventions that will improve human health. So the roles that are available include nurses, scientists, doctors, data managers, statisticians, program managers, admin assistants. The list goes on and on. So you don't have to be a white coat wearer, a scientist, to be a key member of a clinical trial team. So while we were running our phase 1 clinical trial, the UK went into lockdown. And that meant it was very difficult for us to assess if the vaccine was working because people were not leaving their house. They weren't catching COVID. So again, it is only through a network of collaborators were we able to test this vaccine. And some of the key collaborators that we reached out to and called and asked to help us include George Mayanga in Kenya, Suan Costa Clemens in Brazil, and Shabir in South Africa. And through concerted efforts, this global concerted effort, and it really was a global team that delivered this vaccine, were we able to demonstrate that the Oxford AstraZeneca vaccine had a high level of efficacy against mild and moderate disease, and it was efficacy against death of 100%. So 2020 was a bit of a roller coaster for both me and the whole team. I thought I was working hard at the start of the year trying to design the vaccine and help deliver the preclinical studies. The middle of the year was consumed with the clinical trial delivery, and I helped oversee the immune readouts for those clinical trial delivery, the phase 1 all the way through to phase 3. But it was really the end of the year that tested our mettle, that broke some people and was exhausting. And that was the team had to come together, get all the test results amalgamated and put together in such a way that AstraZeneca could do their submissions and we could achieve emergency use authorization by the end of 2020, pre-2021. And it was really in those early months of 2021, in February and March, when we started to see the real world impact of the Oxford AstraZeneca vaccine and other vaccines. And we were able to see that these vaccines were keeping our most elderly, our most frail individuals, care home residents alive. And that was hugely impactful to the team. One aspect that I didn't bank on becoming so important was communication across 2020 and 2021. As I've alluded to, there was a lot of misinformation that was put out about a number of these vaccines. And really, the onus was on the scientists to demonstrate that we were relatively normal people just trying to help. And what I've found is that individuals who are vaccine hesitant or vaccine reluctant, they're generally curious people who have questions that they need answers to. And if you can be honest and frank with them, you can help them reach a decision, either to have the vaccine or not to have the vaccine. And that was something that we definitely took the mantle up, and we definitely did a lot of communication. Having said all of that, and as much as we tried, there was definitely a degree of disinformation out there. This cartoon, this picture on the right-hand side of the slide was a news report that came out that I like to highlight because it's reminiscent of those newspaper reports of Edward Jenner's time, the late 1700s and the 1800s, suggesting that you might turn into an animal after vaccination. Completely untrue, but really hard to tackle. So I would encourage people in the audience to try and have open and frank conversations with individuals that are hesitant, not necessarily individuals who are anti-vax. That's a different type of conversation. And within Oxford, the team received a lot of unpleasant messages, unpleasant support and disinformation. But having said that, we also got very pleasant letters of thanks and support. And this brownie is here to remind me of the individuals that would send us in care packages or would pay for our meals so we could deliver this vaccine. And that was hugely impactful for the team and helped us keep going. The one aspect that both myself and the whole Oxford team and AstraZeneca are very proud of and really wanted to deliver on was supporting equitable access globally. AstraZeneca delivered approvals and emergency use authorizations in over 100 countries, with more than 3 billion doses released to supply in over 180 countries. And approximately 2/3 of these doses went to low and lower middle income countries. We wanted to make this vaccine to get into the arms of people who needed it the most. Your healthcare provision should not be determined by your postcode. And that was an ethos that we shared both within the team in Oxford, globally, and with AstraZeneca. And this vaccine is estimated to have saved over 6 million lives in 2021 alone. Do I think it was enough? No, I think we need to take the lessons that were learned from COVID and do better for next time. The rapid rollout of vaccines in upper middle income and high income countries, there was a significant lag before vaccines got into low income and lower middle income. And we need to enable these countries to make sure that this never happens again, that they are able to make their own vaccines, again, hopefully from the individuals in the audience, so that this scenario never happens during the next pandemic. So what are the types of lessons that I've learned from concept to delivery of the Oxford AstraZeneca vaccine? Well, when a rapid response is required, you absolutely need to have creative approaches to funding and research. And you very frequently need to be able to pick up the phone to your friends around the world and ask them for help. You need to plan for the future and plan many steps ahead. You need open dialogue and collaboration across many governments, the regs of many different countries, charities, industry, and academia. I'm absolutely passionate that we need to build and maintain international capacity. We have local capacity to an extent within the UK, but I'm passionate that we need to underpin international capacity across trials, manufacture, and the rollout of vaccines. And how do we do that? We need to make sure that we have a strong science base in those countries. And I think it's through education and through those schemes that George has already touched on that we'll be able to enable the international capacity. And I firmly believe that we will then be in the best position going forward for a proactive and not a reactive approach. And the critical success factors for me for a proactive approach are definitely first and foremost people and places. So it's you and the audience that we need to invest in, because myself and George are, I don't think we necessarily want to be involved in the next global pandemic. So we would like to pass the baton on to you. We need to have an open dialogue across global collaborators involving government, regs, industry and academia. And we need emergency funding. We need rapid access funding to allow us to respond quickly to these outbreaks. There are a number of filovirus outbreaks that have happened across the last 6 months in Africa. There's an ongoing Marburg outbreak that is happening in Equatorial Guinea and Tanzania. And emergency funding is needed rapidly to deal with these types of outbreaks. But we absolutely also need long-term investment. We can't have short-term investment building up infrastructure or people that can underpin the infrastructure that is only there for 2 or 3 years. We need sustained, joined up, long-term in-country investment. And hopefully in that way, we'll be able to generate impactful clinical research and health outcomes, both for Africa and globally. And then a little bit about me. I thought it might be, how did I end up being one of the advocates of developing vaccines for outbreak and emerging pathogens? In school, I was one of those curious kids that kept putting her hand up. I remember my biology teacher at one point telling me to put my hand down, that whatever question I was going to ask, I'd find out about it in college. I was insanely curious around math, science and art. I was the first in my family to get a degree from university. Certainly a PhD and professorship were not things that my family would have necessarily considered prior to me embarking on my career, but now it's seen as the norm. And it is definitely with the support of my family that I've been able to drive forward in my career. My degree was in pharmacology, not necessarily immunology, but I kept following where my curiosity led me. And that led me to the University of Oxford to work on the immune systems that underpin autoimmunity. And then I moved to the Jenner Institute quite a while ago. And it is through hard work and determination that I've ended up now in the Oxford Vaccine Group, hopefully making an impact on human health and making some good friends like George along the way. So what do I see as my role now? Well, I see my role as enabling the next generation of scientists, enabling you, challenging the status quo, ensuring that what happened during the last pandemic does not happen again, and demonstrating that what we perceive as the impossible is possible and should become the norm. And with that, I'd like to thank everybody who contributed to the Oxford AstraZeneca team delivery of the COVID-19 vaccine, and I'm very happy to take any questions.
Thank you very much for your presentation. I can see people clapping because a very informative presentation. I think what stood out for me was just appreciating what went behind the scenes to ensure that we were able to get the vaccines, we were able to get them to countries like ours. We really appreciate that. During your presentation, you said you don't have to be in a white coat to be a member of a vaccine trial team. And I see a question from Maureen. She says that she is a statistics graduate, passionate about research. Is she eligible to join vaccine research and development, and what are the linkages? I think Tessa, you can go first, and then maybe George, if you have something to add.
Sure, and the answer is absolutely. Statisticians and epidemiologists are frequently and often, there aren't enough of you around to make sure that the trials that the Barmy scientists come up with are delivered robustly and coherently so that we can demonstrate an impact. We have a team in Oxford, within Oxford Vaccine Group, of 10 statisticians that help underpin any clinical trial that goes out of our door. They review it and ensure that whatever we are testing is fit for purpose. So we demonstrate an intervention is good or not good. George?
Yeah, I mean, nothing to add there. It's a skill set that's really critical for vaccine R&D. one of the things that we do as I'm not a statistician, but one of the things we would really need statistical input in is sample size estimations. So how many numbers do you need to vaccinate with your vaccine and with your control product to be really confident with the results that you get? And that's really down to statistics. And it's also down to epidemiology, understanding the burden of disease, understanding where this burden is. Is it in children? Is it in, as I mentioned, is it in adults? And all that information leverages all these different sort of expertise. And that's why I was speaking about an ecosystem. It's not just the scientists in the lab. It's actually down to people who deliver, logistics and manage the supply of vaccine. I mean, these are things that actually can make an excellent product not have impact because you don't have an individual or a group of individuals who know how to deliver vaccines from point A to point B. Over.
I think that's a really good point, George. A vaccine is only impactful if it's in an arm.
Thank you. I'm handing over to Shadrach. He'll take us to the Q&A. Over to you, Shadrach.
Thank you, Professor Lambe, for a very insightful presentation. So there's a question from Joel who'd like to know what if the cell entry factor of a pathogen that is intended for a vaccine design share about 99.8% homologue with the host proteins. So in this case, what other aspects of the pathogen can be considered to select a preferred target?
Yeah, that's a good question. So I think the question is basically asking if the cell entry showed high homology to a human protein, what would you do? So I've worked, my passion for emerging and outbreak pathogens actually came about from influenza, from flu, and trying to generate a universal vaccine against flu. Most vaccines try and target the hemagglutinin, the protein that's on the outside that helps the virus get in. And that changes quite routinely in influenza viruses. But those proteins on the inside of the virus, the nucleoprotein, the matrix protein, etc., and the same with SARS-CoV-2, they have internal proteins. They're generally well conserved, and there are data accumulating suggesting that if we are to target some of those proteins, while you won't stop the virus getting in, you will lessen the severity of the disease. So they would be opportunistic proteins to target if you couldn't target the cell entry factor.
Thank you. So the other question is from Leola from Nigeria, who would like to know if there are internship opportunities in your lab for master's students.
I'll take that as for both myself and George. Yes, there are internships. There are, and George, it might be really helpful if we put together a list of all the kind of courses and internships, and we're able to populate that. I don't know whether you want to speak about AFOX or whether I would just give a very.
I think you can give a general. Just maybe you can talk about AFOX, and then I will invite Dorcas. Dorcas is on the call. Dorcas Mbuvi. To just give a very brief of the IDEAL program, KEMRI-Wellcome. Yeah. But over to you, Tess.
Sure. So AFOX is an initiative to help place brilliant African scientists, like everybody in the audience, to come and spend a short internship with suitable supervisors within Oxford. And I can certainly share the link with that. I can, while we're listening to the next speaker, I can look up the website and paste it in the chat if that would be helpful.
Thank you. So the next question is for. Sorry.
I've just brought Dorcas. She can unmute and make a small comment on IDEAL. I'd seen she had put something on chat, but since she's here, she can go right ahead. Over to you, Dorcas.
Sure.
Thank you. Thank you. Can you hear me, Shadrach?
Yes, we can.
There we go.
Yes, clear.
Yeah, sure. I had already posted. So IDEAL stands for the Initiative to Develop African Research Leaders. It's hosted by KEMRI-Wellcome, where George is based. And we offer different opportunities for students. So attachment when you're in your 3rd or 4th year, internships, especially to do a PGD, a postgraduate diploma in health research methods. So students really learn how to do research. And after this, they can apply for master's studies at different universities or PhDs. And also, we also have fellowships for students who want to do, I saw a question about, do you also fund projects? So somebody has done the taught part, and then they want to do a research. Yes, we advertise these positions, and then you can apply for that. And then you can be funded for a year to do your research with us, your MSc research, and it can be in vaccinology or other subjects like immunology, clinical science, epidemiology, health systems strengthening, so different themes. Also, we have an MSc at Pwani University. We collaborate with Pwani University on immunology, and another one on bioinformatics, especially for students who have done stats and they want also to tie it up with the biology and also statistics. Here we are collaborating with the University in South Africa. We also have fellowships in for students who want to do PhD. So once you're done with your MSc and now postdoc after that. So we advertise these positions regularly on our career website, and I've posted our career at the jobs.KEMRI. And then please apply online. And every time we post everything, we also tweet it. So please follow us on Twitter, and you will for sure be informed of all the opportunities once they come in. Over to you, Shadrach. Joy. Thanks.
Thank you. Thank you, Dorcas. George, there was a question on whether challenge studies are done in humans.
Oh yes, they are done. Yes, they are done in humans, and in fact, you'll be pleased to know that Kenya is one of the countries that's leading in this area. So at the KEMRI-Wellcome Trust Research Programme, we're able to do, they're called human infection studies now. So where you expose in the same way that I described with the animal studies. So you would expose humans to a pathogen in a controlled manner. And observe the sort of natural course of that pathogen, whether you want to study the clinical manifestations or you can even test a vaccine. That's the aim. But there are some key criteria that need to be fulfilled. You need to have a rescue treatment, as we call it. So you can't expose somebody to a pathogen for which you have no treatment. So malaria, we do have treatment, so that works well. And the other challenge agents such as Shigella. So it's an area that will develop, and I know there is dengue as well in other countries. So the answer is yes.
Thank you. Yeah. So one further question, as we keep the questions coming from the chat box. So vaccinology is a specialist subject that is not taught at the undergraduate level. So how do we bring on board decision makers from the tertiary level of education, say university, senate, administration, etc., to get them thinking about vaccinology and to hopefully get students talking about vaccinology as a career?
Right. Okay. Over. I'll check that from what I see, that like at least from our context, what at least a strategy we are pursuing, and Tess can come in with a UK perspective. I think for me, it's all about awareness, just awareness, like what we're doing now, awareness of what it entails to make a vaccine and the impact of vaccines and the utility of them for not just human health or animal health. It's also the economic, it has an impact on economics because vaccines were deployed for COVID. Countries then opened up and there's more travel. That has a real impact on economics. But it's also just having examples and really promoting when you're in situations where you can advocate for a particular tool to a situation, and now I'm thinking about health, really articulating, communicating these benefits of vaccines and the value they bring to society, to this fora, because I think that's the only way things are prioritized at a very high level. and from then on, it permeates through. But just that increased awareness, like what we're doing now, I think, and on a massive scale, and the ambition here is to do this across the continent. I think that will already start moving things in the right direction, because there are many other activities happening around here. It's just that they need to be brought together in a way that's impactful, and people need to speak to each other around this. But over to you, Tess.
That's a really good point to end on, George, with the people need to speak to each other. I'm a little bit sick and tired of people remaining in their little silos and not communicating, so completely agree with that. I would have to agree that even within the UK, I don't know that there are bespoke courses that are done at undergraduate level in vaccinology. You're generally introduced to biochemistry or biology, or in my case, pharmacology. And you need people like George or myself, people that you can relate to, that you can see almost yourself having a similar career path. And really, it's about your curiosity driving you. There's no point in you being stuck in a career that you hate, that you don't want to do. And there are lots and lots of opportunities within both academia, industry, manufacturing, clinical trial delivery. So, for example, we will have people who have gone through their undergraduate, have done their PhD, are in the lab and suddenly decide they don't want to do that anymore. And that's okay, because they can still stay within the clinical trial ecosystem and become project managers and ensure that the trials do get delivered. And actually logistics and that mindset of, well, if A happens, B, C, and D need to happen is really important. So it's about a mindset of wanting to give it a go, being okay with being challenged, and a lot of hard work.
Thank you very much indeed. And as part of that follow-up, I was just wondering whether there's opportunity to leverage on online learning. So for example, teaming up with massive and open online learning platforms to deliver vaccinology short courses as part of the 3 strategies that George spoke about.
So it's interesting that you raised that. I was just typing up on the channel and I posted something there, which is a collection of quality assured vaccinology courses that at a global level. There are very many courses listed there, and do have a look at them, and because it will begin to sort of provide some exposure to the kinds of things that a vaccinology curriculum entails. And you can already then start seeing what gaps you have in your career so far, what things you're interested in. I went into vaccinology because I wanted to do something translational. You may already be doing something very translational. I was working on a very complicated molecule called PFMP1 in malaria. So it's like the most diverse thing I've ever heard of. But it was intellectually stimulating. I really loved working on that field, but I wanted to do something different. So at that point, I knew what I did not want, and I was very clear on that. I wasn't very clear that I was going to work on vaccines. I knew I wanted to do something translational to provide a solution for the kinds of things that I talked about. But looking through some of these courses, some of the resources that are available now, and there's more now available than at least from at the time when I was considering this, you can already begin to see where you fit in this sort of ecosystem. And I think that's the first step. And then you've got that awareness, you've got access to information. And from then on, then it should be straightforward, I think. At least throughout this discussion, there's been lots of mention from myself and Tess and even Dorcas about potential opportunities. And these are just the opportunities we know of. There are very many out there. So I think I would just encourage you to do some digging, let those fingers do the walking and through the internet and try and identify opportunities out there, because there are very many. Over.
Thank you. And then there's a question around breakthrough of HIV vaccine. Where are we with that? Professor Lambe?
Yeah, I'll go for that one. I'll say that's a tough virus to make a vaccine against. I have to admit, while it doesn't feel like we were very lucky with SARS-CoV-2, we were, because we could make a vaccine. If it had been a pathogen like HIV, I think we'd be in a much worse situation. HIV and other types of viruses that are similar, they're really good at rapidly mutating and have lots of different ways of escaping the immune system. I know we have variants of concern, and I know SARS-CoV-2 does mutate its spike protein, but the immune system, the immune response that we lay down at the first exposure does seem to temper disease. And we're not seeing ICUs being filled up rapidly like we were when we were completely naive. So I'm hopeful for a HIV vaccine, but I'm an optimist and have been hopeful for 20 years. So if I leave it at that.
Thank you. Thank you very much indeed for that. So there are no further questions on the chat. Joy?
Thank you, Shadrach. Do we have any other question? Is anyone interested in raising their hand and asking a question? Okay. So just give me.
Yes, there's Leola there.
Okay. So we can start. I'll just give you. I'll enable your mic so you can go ahead.
Yes. Can you hear me?
Yes, we can.
Okay, thank you, Professor Lambe and Professor Warimwe, for your presentation. It was very insightful. So first, I want to say that I'm currently studying vaccinology in Europe, and as part of my program, I have the opportunity to conduct a 4th semester internship at any partner institutions. And that's why I asked the question, because one of the partner institutions is the Jenner Institute of Vaccinology at Oxford. And I've been checking the research at the University of Oxford, including the research of Professor Lambe, and I wanted to know if there are internship opportunities available, so I could put up an application. And this is supported by my program, which is the Erasmus Leading International Vaccinology Education Program. And it's part of the program that is listed in the link that was sent by Professor Warimwe as well.
I would say that myself and George have too many ideas and never enough good students. So, and we're always coming up with new ones. Yellow fever springs to mind, George. But please do just reach out to me. I'm very happy to have a conversation about the types of opportunities we could have.
Okay, thank you so much.
Thank you very much. Noni, I saw you had your hand up.
Sorry, no, I was trying to do some other things on the screen. Sorry about that.
It's okay. Shadrach, I see you have one more question on the chat. You can take that and then we can wrap up the Q&A session.
Yes, thank you. This is a question from Inez Odhiambo, who says varieties of vaccines have been developed for the control of viruses, but viruses have got no cure. Yet some vaccines have been of positive impact. So what is it that has been the problem encountered in the manufacturing of vaccines that cannot allow it to completely heal the patients? And then what is the key problem with the viruses that makes it impossible to manufacture their antidotes? Professor Lambe?
I was wondering who you were going to go to there. So lots of viruses work in different ways. So some viruses, so herpes virus, for example, they lay dormant in your system. You don't ever clear them very effectively. And that is one of the key problems with viruses that can make it difficult because they hide to manufacture vaccines that will completely eradicate them. And manufacturing of vaccines that cannot allow to completely heal the patients. I'm going to be a little bit cruel here and say that if we, myself and George, make vaccines that keep people alive and keep them out of ICUs, that a small degree of infection or discomfort can actually be okay, because it can mean that your immune response towards that virus after vaccination is boosted. So once we're not making people overtly sick with repeated infection and helping boost the immune response and keeping people safe and out of hospitals, vaccines that prevent complete infection and complete disease, while they are the golden chalice, or the goal that we all aim for, they're not necessarily always the best. George, anything to add?
I think, yeah, you've covered that, but I think I'll add something on manufacturing and difficulty in manufacturing, and I'll use an analogy. I think we've got, if you think of something like, and I'll use food. I mean, it's almost evening, maybe I'm hungry, but I'll use food as an example. So pilau is a common, food, especially in coastal Kenya. I mean, we can think of also like jollof rice as an example. So if you make one of these dishes in your home, and you follow the recipe very well, and you have guests over, and the food tastes really nicely, and you've done a great job. It's a fantastic dish. Now think you need to make that specific meal with all those sort of specific ingredients and to taste exactly the same for your village. Think about how difficult that will be. You need bigger pots, you need, the ingredients, you need to weigh them up, the water volume, you need to think about lots of different ingredients. Now scale that up beyond your village to the country, to the region. Just think about how the complexity of doing this at a global scale. And when you're manufacturing vaccines, you're not just manufacturing vaccines for your neighborhood, for 20 people or for 100 people. You're thinking about millions. Otherwise, that vaccine enterprise is not going to be sustainable. And some of those complexities are worse for some pathogens than they are for others. And that's why, you can have a fantastic idea at a concept level, do the initial immunogen design and everything, and it looks good on the computer. It looks like, wow, this construct will work very well. And then you get to manufacturing and it fails. So that's a key consideration, and you highlighting the sort of difficulties in manufacturing is actually one of the key bottlenecks of vaccine R&D, for why some pathogens or some vaccine constructs actually do not advance. They are not scalable. You can make them at a small scale and they look fantastic. You can use them in your proof of concept studies and they look very well. But then when you move on to scaling up the manufacture, it's impossible, either because of the technological difficulties, or if you can do it, it's extremely expensive, which makes it untenable as a construct, for deployment, as Tess mentioned earlier. But I hope that answers your question. But it's a very good one.
I just add that you're making me very hungry, George. That's all.
Yeah, I was gonna say thank you for whetting our vaccinology appetite. Well, over to you then, Joy.
Thank you. I see we have one more question because we have time. Marianne, you can go ahead and ask your question. Just unmute. I've given you the rights.
Okay. Hello?
Hello.
Hello, can you hear me?
Yes, we can.
Okay, thank you. Thank you very much, Professor Lam and Professor George Warimwe, for the great presentation. I'm not sure if you addressed the question I'm about to ask. I had some problem with my internet connection, and it's based on the side effects caused from what do you call it? From the phase 1, phase 2, phase 3 trials of vaccines. So I am a graduate from the University of Nairobi. I studied biology and I have an interest in advancing my career in vaccinology. And occasionally I share articles about malaria vaccine research with my friends. And one of the debates we have is about the side effects that the vaccine causes. So I wonder, how do I explain? One of the side effects is there were deaths that came from that arose from the phase 2 trials, I believe, of the RTSS malaria vaccine. But then I tend to believe that sometimes if you look at the number of, deaths that arise from these cases against the successes of the trials, then it's safe to say that there could be other underlying side effects. So how do you explain to them that sometimes the number of deaths that arises is small and maybe negligible compared to the success of these phases of the vaccine?
Do you want to take that, Tess?
Sure. Well, I'm going to pass over to malaria with you because you definitely know more than I, but I'll talk about rare adverse events. You've actually answered you've answered your question somewhat in the way that you've presented the data. There's generally a number of deaths that will happen anyway because of random events. And if the recording of those deaths because of an event is at the background level, if it's not above what normally happens, those deaths will still get registered because those individuals are part of a trial, but the number of deaths will not be above the background level. That could be one explanation, but I don't know about the data for the RTSS. I will discuss the very rare adverse event that was seen with the viral vector platforms with Janssen and with Oxford AstraZeneca, TTS. And those very, very rare adverse events were not seen in phase 1, phase 2, phase 3. They were only seen when the vaccine was in later stages and it was out and being globally distributed. And actually what we've tended to see now is that there seems to be an association or a possible association with a genetic predisposition in certain geographies. We're not getting reports of the TTS in large parts of the world, whereas we have had more reports in Northern Europe. When you know about these very, very rare events, you can start to develop therapies and you could start to develop interventions to prevent them from happening. And you really do have to do, and it's not a simple maths equation, but you have to do a weighted analysis of how many deaths will the vaccine prevent as opposed to a very, very, very rare event. And those are the types of calculations that nobody enjoys doing, but have to be considered. George, anything you'd like to add?
I think you've covered that very well. I mean, the only other thing I can mention is goes to your first point. Like if you take, 10,000 people, Marianne, and follow these 10,000 people up for a year, just record, what they are up to, and for a year, there might be some who will die, some will come down with colds, and some who will get other illnesses or other experiences, and that's the sort of background level in your population. Now, if you take a different 10,000 people, like at the same time, so you started with 20,000 and 10,000 are given a vaccine or a drug or any other intervention and monitor. So, you then want to compare the incidence of those two things in those two populations. And what you're describing there with the RTSS vaccine trial or use of the vaccine might be one of those sort of incidences. I'm not really familiar with a particular case you're talking about. But this is the thing you have to think about. And this is where then going back to statisticians, you need to look at, is this incidence above the expected background rate of the population? So now you start bringing a different discipline, because then the statistical input to that gives you the confidence to say, okay, well, this is more than is expected. And then you can then do something about that in terms of that vaccine program. But in the same breath, as Tess has mentioned, in terms of effectiveness, you can then start looking about the cost benefit and impact on the health system. Again, then there you start bringing in not just statistical modelers, but also health economists. So there is space for a very broad group of expertise to sort of make these decisions about the use of interventions, because it's always a sort of a balance between the benefit and any harms that may arise or any cost that may arise, and these are multifaceted. They're not very simple. So you have to look at the whole picture. Over.
Thank you, everyone. I think we've come to the very end. Before we go, I'd like us to fill in the post-event survey. I've put it in the chat. And as we do that, I'd kindly request Alan. Alan is participant, and he's been engaging with the program for many years through our different university engagement programs. Alan, you can go ahead and unmute. He would like him to give us a feedback and reflection on how this session was. It's more like a thank you note to you, George and Tess.
Hello, everyone. Thank you, Joy, for this opportunity to, be the final person to speak in this webinar. I really don't take it lightly. First off, I would like to thank the organizers of this webinar. It's really been an interesting webinar, considering when Joy first informed us of this webinar, I really was interested in actually getting to understand what will be dispensed in this meeting. And I'd like to say that my expectations have been met. In fact, you have exceeded my expectations in this webinar. Therefore, thank you so much, Professor George Warimwe and Professor Tess. I guess my take home from this engagement is that you have to be very proactive, and especially on my part, I have to be really proactive in terms of looking out for these opportunities in career development, and especially in vaccinology. So thank you so much. And interesting to note is that you had actually begun designing a coronavirus vaccine even before pandemic. I mean, that's ingenious to say the least. Therefore, thank you so much, Professor Lam and Professor George, for your engagement and taking your time to, tell us and show us the work you have been engaged in all this while. It's really interesting, and I really can't wait for the next engagement. I really also hope to see you in the next engagement if you will have the privilege to attend. Therefore, thank you so much. Thank you, Joy, as well, and also thank you, KEMRI-Wellcome Trust, for these engagements. They're really exciting. Thank you all. Have a good day.
All right. Thank you, Alan. Before we leave, I'm still waiting for you guys to fill in the survey, and we have 12 minutes to go. I don't know if Noni, you'd like to make any comment before we come to a close. I don't know if she's in the call. Okay, she might not be on the call. That's okay. So thank you everyone for joining us today. I will send you an email with the information on IDEAL, as well as the course that George had shared after this meeting. And also I should be circulating posters for upcoming webinars once we have this done. Thank you everyone, and have a good evening.
Thanks everyone. Have a good one.
Thanks. Bye-bye.
Bye.
Thanks,
Bye-bye.
bye.